Well, it's a real pleasure to be here. I was last in Fribourg 47 years ago. I was living in Bern, you saw it up there, and I came as a tourist and spent a weekend in Fribourg. I've been through it on the train, but never stopped before, so it's a pleasure to be back here. And it's a pleasure to be giving, am I not on? Did you hear what I just said before? So I don't have to say it again, no? Right, okay, good. And also to give the Weizmann lecture, I've been to the Weizmann Institute, where he's buried in actual fact. He had close linkages, as you've just heard, in the UK, in Manchester, and even closer linkages with Israel, of course, as the first president. He became the first president, as I just noticed there, 2 weeks after I was born, I saw. So I thought I would give a talk tonight, not about research in my lab, but a more general talk, and the title of it is, What is Life? So the first thing I'm going to say is I'm not entirely certain that I'm going to give you a satisfactory answer to the question that I've set myself, but I will have a go. And I hope you'll find something out. Let me just push that back. I hope that you will find, it's all right, don't panic. Yep. That you'll find something out about living things that you hadn't thought of entirely before. Have I pushed it off the end here? Yes. Is that looking stable from your point of view? Good. Well, I shall hold this here. Good? All return. So, what is life? It is a fundamental question in biology, a very easy question to ask, not so easy to answer. And why is that? Well, we start with the fact that there's a bewildering diversity of life on our planet. All around us there's bacteria, fungi, insects, animals, plants, and of course ourselves. We're all physical entities, but physical entities that are unusual and strange. Entities that can maintain themselves, grow, self-organize, and reproduce. And the question that I want to try and address tonight is, what is it that links us all together? And I put up here a butterfly as a sort of fairly simple life form. Actually, a rather beautiful one. For those who know about butterflies, it's called a yellow brimstone in English, and it comes out in spring. An example of something that is alive. And the question is, what is it that links that butterfly with us? What do we share in common with a butterfly, or even beyond that, with these plants, or even beyond that, with a microbe? And by thinking about that, we can perhaps unpick what are the principles that underlie life. So that question, what is life, is one that's been asked for many years. In fact, this is the title of a book written by the very distinguished physicist Erwin Schrödinger, which he wrote in 1944, as you see here. And it was very influential at the time, including for me, which I read it about the same time as I came to Fribourg, so about 50 years ago. And I'm going to quote from it a little bit later. I'm going to ask the same question as he asked, but I'm going to take a different approach. I'm going to consider some of the important ideas of biology, and I think us biologists don't talk enough about ideas. You know, if you go to a popular, the book in a bookshop, the shelves of popular science, you'll see lots of books by physicists talking about great ideas, you know, relativity, quantum mechanics, and the like. But us biologists tend to write about what is going to be the next medical breakthrough, which usually doesn't happen, by the way. rather than focus on really what are some really fantastic ideas. So I'm going to explain to you what I think are some of the important ideas, 5 actually. I'm going to explain what they are, I'm going to explain where they came from, I'm going to mix it up with a bit of history and a bit of philosophy, and hope to develop some answers to this question about what is life at the end of it. So the first idea is this one. It's the concept of the cell, which we certainly don't talk about enough. Because the cell is the basic structural and functional unit of life. It's life's atom, as I've said up there. Now, they were discovered by a scientist in 1665 called Robert Hooke. And here is a picture of the microscope, or rather a picture of a microscope that would have looked a bit like his microscope. And there is also an image of these, what he called cells. And what he did is he took a razor, cut through cork, looked at it under the microscope, and saw all these boxes here. That's the black and white one. I put next to it a modern scanning EM, just to show you it is exactly the same. He saw these neat arrays of boxes. He called them cells, after the Latin word cella for small cubicle, and he published them in a book called Micrographia. It's actually a very expensive book today. If you get hold of one, it's worth 100,000 Swiss francs, by the way, just to put it in perspective. And this is the first picture of a cell that we have. I collect old books, actually, of science, and I have a book that was published 7 years after, which also has pictures of cells in it, but it's not worth 100,000 Swiss francs. A few years later, a Dutch draper, a sort of artisan, Anthony van Leeuwenhoek, working in Delft, scraped between his teeth and discovered single-celled life, discovered bacteria. He sent this drawing I'm showing you here to the Royal Society in London, and he sent actually over 1,000 letters to the Royal Society, which had only been around for about 20 years then, recording what he'd seen. And what you see here are the pictures he saw when he'd scraped between his teeth. I particularly like figure BCD, where it's obviously done some sort of loop-the-loop under the microscope. He notes he was very disturbed by what he found between his teeth, because he was rather proud of his dental hygiene. And to find little animals living in it was very, very disturbing. Now, he lived in Delft, and he was a close neighbor to the painter Vermeer. Now, Vermeer, who many of you will know of, was unusual. He only painted about 30 to 40 paintings that we know of today. 2 of them are of a scientist. It's the same scientist. Here's one of them. It's in the Louvre. And I like to think this is Leeuwenhoek. They lived in the same street at the same time. There is 0 evidence for my speculation. But although when Vermeer died, Leeuwenhoek was his executor of his will. So there could have been some relationship between them. Over the next couple of centuries, 2 centuries it took, it became clear that all living organisms either are single-celled or a collection of cells. And that was summed up by Theodor Schwann, this rather fierce-looking German professor from the mid-19th century, who concluded, together with a botanist called Schleiden, we have seen that all organisms are composed of essentially like parts, namely of cells. So the cell is the basic structural unit of life. But as well as that, they're also the basic functional unit of life. What do I mean by that? Well, this was stated by another rather serious German professor called Virchow, who was, by the way, the founder of pathology. And he, in 1858, a few years later, said, every animal appears as a sum of vital units, each of which bears within itself the complete characteristics of life. In other words, he was arguing that, that cells are like living organisms. And I conclude from that, if we want to understand what life is, then the simplest entity that we should think about is the cell. So I'm going to focus on the cell for the rest of this talk, or for most of it. He also had this Latin sentence, omnis cellula acellular, all cells come from cells. This was quite important because at the time it wasn't certain whether they came about spontaneously, but what he was arguing was that cells come about from the division of pre-existing cells. So, excuse me, I've turned over too many pages here, and I shall leap forward a century or two. Now, I hope that you're finding cells interesting, because what this means is that there's a stream, I want to emphasize it, there's a stream of connection through cell generations which stretches back through the development of individual animals and plants, and then extends beyond that into their parents and their parents and their parents into deep time during the evolution of life on our planet. In other words, there's a connection that just goes back through an individual and then through that individual back to the parents of that individual and so on. And that's something just to think upon that we don't generally think about. Now, have I convinced you cells are interesting? Yes? If there's any doubting people, I'm going to show you this. This is a sperm being, this is an egg, sorry, being fertilized by sperm. It's a mammalian egg, but it looks pretty much the same as, it's a mouse egg actually, but it looks pretty much the same. At least I was told it was a mouse egg, so I believe that. It was pretty much the same as a human egg. And I need to remind all of you in the audience that every one of you once looked like that. Every one of you was once a single cell. And if you're not interested, there's a door there and there. Now, I'm arguing, therefore, and it's generally argued, of course, that cells are the simplest entities which exhibit the characteristics of life, and that means understanding cells, as I said, is key to understanding life. Now, what can we say about cells? Here's a modern picture. They are bounded physical entities. That sounds a bit grand. It becomes relevant to say it's a physical entity when we briefly will mention computer programs that have some life properties, but this is a bounded physical entity, separate from its surrounding environment, but in communication with that surrounding environment through a semi-permeable lipid membrane. So it allows them to be independent of their environment, and yet in communication. Why am I stressing that? I'm stressing it because when physicists think about this problem, I know I'm beginning to sound as if I'm obsessed with physicists. My daughter is a professor of physics, so I have to keep mentioning physics or I get into trouble. But this separation means that cells can become more ordered and complex at the expense of increasing disorder outside that bounded entity. And in this way, of course, it doesn't contravene the second law of thermodynamics, which worries physicists. They don't have to worry about it because it's separate from the environment and you can get that complexity. Schrödinger was astonished by this, and here's two quotes from his book. The first is, he noted the organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. They write so colourfully, these physicists, don't they? You can see why it's in the popular section of the bookshops all the time. Thus escaping the decay into atomic chaos. But there isn't a problem with the second law of thermodynamics, so put that aside, although sometimes people worry about it. He also had a brilliant sentence. Chromosomes, he was talking about chromosomes, which are the seat of heredity. I'll say something about that a little later. Contain, he said, some kind of code script. The chromosomes contain in some kind of code script the entire pattern of the individual's future development and functioning. In other words, there's something about the chromosomes which encodes or certainly influences the development and functioning of a cell and an organism. And I want to turn now to my second great idea, which is the gene, and trying to explain the really fundamentally mysterious phenomenon of inheritance that Schrödinger himself was mentioning. Now, this is the idea of the gene, which had its origins with the Augustinian monk Grigor Mendel, about all of you have heard. He was abbot of Brno University, now in the Czech Republic, then in the Austro-Hungarian Empire, and he published his two papers in 1865. Here he is. He became abbot just a little bit after this. I find this quite interesting because I've visited the monastery quite a few times now, and it was actually like a research institute. Now, we don't think of monasteries as research institutes, but they were. They were there. They were essentially academics carrying out research and often teaching in the local environment. And this was going on in parallel, if you like, to the traditional university system that we are familiar with. So Grigor here, he tackled the problem of inheritance by studying plants. He actually did preliminary experiments in about half a dozen different plants. Most of the crosses he did, he couldn't make any sense of, and rather sensibly abandoned them and chose to focus on peas where he could make some sense. Now, some people would say, well, that's just cheating, okay? But in actual fact, he was very wise. What he was saying is biology is complex, and if you're trying to understand it, you're best trying to choose something that you can understand, and by understanding that, you might then be able to apply your thinking and your principles to those that you don't understand. And we have to think about that in biology, in my view. And what he did is he carried out many crosses of pea plants, which had different characteristics, and then analyzed the outcomes of the thousands of progeny that formed in these crosses. And frankly, he was quite well equipped as a scientist in this monastery. This was his garden. I visited this in 1981, height of the Cold War. I got this picture then. And he had a greenhouse of the same size. The monastery built him a greenhouse so he could do his experiments. I mean, completely extraordinary. And what he did is he crossed peas of various characteristics. You see some of them here. Which included plants of different heights, flower colors, seed shapes, and so on. And famously observed, and I'm not going to go into the details of it, simple ratios that came about in the numbers of the characteristics that were produced by, of the peas that were produced by these crosses. Famously, ratios like 3 to 1 that we all learned at school, or probably most of us learned at school. This eventually led, not actually through Mendel, who only partially grasped what he was doing. It took another 30, 40 years, during which time his ideas were completely forgotten, by the way, when it was rediscovered by 3 other botanists doing similar crosses. And this eventually led to the idea of what we call Mendelism, which is that heredity is based on indivisible particles or factors that are inherited through the germ cells, ovules in plants, sperm in eggs in animals. And it's the segregation of particles that give these fixed ratios. And so that was the brilliant piece of reasoning. So then you start thinking of Schrödinger's code script, which of course was later than this, in terms of particles being passed on from one generation to the next. These factors were found to be linked intellectually, initially, to chromosomes, which were discovered a little bit later. I picked this picture of 19th century chromosomes, and you in an onion tip. And I have to say, I first saw cells and chromosomes when I was at school, and it just looked exactly like this. We took the onion tip, squashed it, and saw it. It was just this. You know, it could be exactly this. And it looked as if in the dividing cells, chromosomes were separating out, and it was speculated that this was the basis of heredity. And these factors are what we now call genes. And genes were shown to be made up of deoxyribonucleic acid, or DNA, by Oswald Avery, working in Rockefeller University in New York, not by Watson and Crick, we'll come to them in a moment, but by a biochemist, Avery. And I was in Rockefeller, I was president, in fact, of that university. And he had a junior assistant called McCarty. They published this paper in 1944. And when I went there in 2001 or 2, this little old man came up to me to welcome me to the university. And he said, "My name's Matt McCarty." And I looked at him and I said, "You're not the Matt McCarty, are you?" And he said, "Yes, I am the Matt McCarty." And he was still working there, and he published his paper in 1944. Anyway, Avery suggested that genes were made of DNA. He was hugely criticized at the time, mostly by his colleagues, biochemical colleagues in Rockefeller University. You know what academics are like, I'm sure. But he turned out to be right. Now, what Watson and Crick did, based on data from Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, was to determine the structure of DNA. And Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases that make up DNA. And I'm going to talk a bit more about that a little bit later. So I'll only say a little bit now about the structure of it. And here you see the chromosomes unraveling to reveal the double helix, which is more traditionally seen here. And DNA, you can consider it like a twisted ladder. And the rungs of the ladder are made up of the links between the complementing bases on the two strands. And because those complementing bases pair with each other, an A with a T, a G with a C, let's not worry about what they're called, this can explain how the molecule can be copied during reproduction. So this was a very exciting discovery. Crick went on, together with others, to propose what he called the central dogma, and it's shown here, where the sequence of a gene, a DNA, which is at the top there, is copied into messenger RNA, and the messenger RNA is then translated. That's a process of transcription, because it's just basically a copying, translated into protein. And I'm going to talk again a bit more about that a little bit later. These discoveries were made in the mid-20th century and a little bit later, and they laid the foundations of what we now call molecular biology. Central to DNA is the genetic code, and heredity is written in this 4-letter code of A for adenine, T for thymine, C for cytosine, and G for guanine. And this way of storing information is the same as my speech. It's the same as sentences in a book or bytes in a computer. It is linear digital storage of information. And again, I shall return to that. So that's two ideas. The third idea is life as chemistry. And that means that you can understand life, in some senses anyway, in terms of chemical activity and physical forces. And this wasn't always recognized as the case. It was thought until about the mid-19th century. And you know, something that's a bit odd, all these fundamental ideas were all sorted out by the 19th century. I mean, what have we been doing since, I ask myself? But the really great ideas are often very old ideas. Puts us in perspective, I'd say. The origins of this idea can be traced back to French chemists, actually. Initial work by Lavoisier, the father of chemistry. And is he over there? I couldn't quite see. Is he the one on the left? Yeah, okay. And on this side, we've got Is that Mendeleev or? Ah, yeah. Okay. Very good. All right. Thank you. Thank you for that. Right. So here he is, Lavoisier, who carried out experiments as being French on the fermentation of wine, and was playing with the idea of chemical transformations being linked with life's processes. He didn't get to a conclusion there because, unfortunately, he was also a tax collector for the ancient regime, and he lost his head in the French Revolution in, I think, 1794. Famously, his judge said something like, "The Republic has no need for savants and chemists." Savants and chemists. I always say we should beware of politicians, by the way. It was a political court. However, he was picked up, his work, by Louis Pasteur, another French chemist, here he is, who was also analysing fermentation. What is it about these French? Who was also analysing the fermentation of sugar beet to make alcohol. And he was doing it at the request of industrialists in the north of France, because they were carrying out fermentations to make alcohol. And they quite often failed, and they turned to Pasteur to say, could you explain this? This is absolutely applied research, when people make, arguments, there's pure research and there's applied research for the, for the mean mechanicals. He was asked to investigate this, and what he discovered is when the fermentations worked, they had yeast in the fermentations, and when they didn't work, they didn't have yeast and seemed to have bacteria. And he said, okay, you can solve, industrialists, your problem by just making sure you've got yeast there, and then it always will work, which apparently was the case. But what he then reasoned was these different cells were making different chemicals. One was making alcohol, yeast. The bacteria were probably making the acid. And so that was a different sort of outcome. I realise I've got experts on bacterial fermentation in the audience, and I'm getting really nervous about that. But anyway, I'll move on. But he then concluded more generally, and this is the brilliance of Pasteur. He made the observation and then thought, what does it mean? What is the idea that could be revealed here? And he concluded, and I quote, translated into English, more generally, that fermentation was a physiological act yielding products for the cell. Therefore, chemical reactions are an expression of the life of a cell. Quotation in the 1870s. That's the foundational statement of biochemistry. And it came from a study of fermentation in an applied project in the fields of northern France, looking at sugar beet fermentation. And what he was concluding is life is chemistry. But it's a very elaborate and special chemistry. It has its basis in carbon polymers. We've met carbon polymers in DNA and RNA, which I've briefly mentioned. But now I want to consider the carbon polymers that make up proteins, because they are central to life. Carbon, by the way, has its origins in the centers of stars, dying stars. So we literally have our origins in the stars, which is something else really sort of quite nice to think about, I'd say. And the substances from yeast that Pasteur was postulating were responsible for the life of cells are proteins that act as enzymes that catalyze chemical reactions. That's where he didn't discover that. That was discovered mostly by French biochemists, it has to be said, between the 1870s and 1900, whilst he was inventing vaccines and things like that. Proteins are polymers. They're polymers of amino acids with a big focus on carbon, but also nitrogen, oxygen, and hydrogen. And the carbon atoms form a part of the backbone to the polymer, and they link side chains of the amino acids. So you can see that here. If you look at the bottom line there, that long line, you see carbon and nitrogen, which is making up the backbone, and then what you see are different amino acids, which are side chains. I've written it out like this so it's emphasized. They don't actually look quite like this. And you'll see that they are very different. And they, as a consequence, have very different chemistries. Some are positively charged, some are negatively charged, some are hydrophobic, some are bulky, some are not bulky. And these are examples of some of the 20 different amino acids that make up proteins. And this is a zoom in into that backbone structure that I just showed you. And if you didn't notice it too much, there you see, and here it comes out, we can see. Now, what is the consequence of this? The consequence of this is that there's a wide variety of different chemistries that can be produced by these proteins. This isn't the case for DNA or RNA, which is rather boring with only limited chemistry. But proteins can produce elaborate structures and which are capable of carrying out very many varied chemical reactions. DNA is dull, which is why, by the way, Rockefeller's colleagues thought, not Rockefeller, Avery's colleagues thought that he was wrong, because it was too boring a molecule. But the chains of amino acids that you see here can form these longer chains, because proteins are often hundreds of different amino acids. And these, because of their different chemistries, fold up in a wide variety of different ways, and they expose different chemistries to the environment as well. And this is an absolutely beautiful system for producing elaborate chemistry and a wide variety of chemistry. And it all just falls out of the nature of the amino acid polymers that they're made up. So these enzymes and proteins, they're acting essentially as chemical machines. They produce a huge range, really, of the chemicals required for living organisms to function. They can make new molecules, they can break molecules down, they can recycle them, they can generate large molecular assemblies, which can make the other polymers of nucleic acid, lipid, carbohydrates, as well as protein. They can also do physical work. There's a lot of chemistry going on, and that's central, but they make motors, they carry cargoes around the cell on protein-based railways. They act in a variety of hybrid ways, chemistry and physics. They can capture energy from sunlight. They can break down sugars to produce energy that can necessary to drive all the activities in cells. I'm not being specific, I'm trying to make you think this is wonderful, because it is absolutely wonderful what they can do. And a consequence of it, if you think about it, is all this activity of thousands of different chemical reactions are being carried out simultaneously and close to each other in tiny single cells of micron dimensions, or a few micron dimensions. And you can put all of this together. This is a metabolic map that some of you may be familiar with. It's just the central Krebs cycle and associated metabolic pathways. And each of those dots is an enzyme. And this is only a small fraction of the chemistry that's going on in cells. Now, how is that possible? How is it, if you're a chemist, you create little microenvironments to allow the chemistry to occur. Here, what a living cell has done is created thousands of reactions simultaneously close to each other. And that is really only comes about as a consequence of compartmentation. And that is critical to what I want to communicate here to you, because it brings together some of the different things I'm telling you about. That compartmentation is brought about by the surfaces and folds of the proteins that make up the enzymes, complexes of different enzymes acting together, by membrane-bounded organelles that also compartmentalize activities, and is the basis of what we see here in the cell. Now, I'm now asking you to look at the cell again and not think of it as just a structural unit of bits. What this is, in fact, is thousands of different chemical microenvironments, all linked together, carrying out a hugely elaborate chemistry that is the basis of life. So don't think of it as just something, you know, as a structure. It is actually elaborate chemistry that can only occur if it's compartmentalized. But, I told you this is wonderful, but it's got to work together as a whole. It's got to be connected. It's got to be regulated. Now, how can that be organized? Well, that's only possible if there's communication between these different environments, chemical microenvironments, and organization so it can act as a whole. And that in turn means that there has to be a transfer and organization of information within the cell, because the organization is based on a management of information. And that leads to the next idea. I've talked about life as chemistry and, of course, physical forces. But life is also information. It's based on information. And the concepts here are complex systems, control, and even this word that us biologists are very wary of, purpose, of acting as a whole. Now, life is a complex system. Do you know, I looked into seeing where was that first stated, and you're going to be astonished with the answer. The first part where I found where it was stated was by Immanuel Kant, the philosopher, early 19th century. He argued, because he was thinking about living things, in particular humans, and he argued that they are complex interacting systems. And he wrote about 20 pages on this in his book on moral philosophy. Why he did it, I haven't the slightest idea, but it's in there, and it's pretty erudite if you read it. And to get the operation of a complex system, you need to manage the information flow within that system. And I'm going to give you two examples of how information is important in the life of cells. The first is to go back to the double helix of DNA, and this is the obvious one, and you'll mostly know what I'm going to say. The structure of DNA is iconic and beautiful, although I don't want to go into another research institute and see a piece of art based on a helical structure. It drives me to complete distraction because it's completely overused, but we'll pass over that. But actually, its real beauty for me is not this twisted structure. It's because it can only make sense biologically when it's recognized that this is, in fact, a digital information storage device. I mean, that's what it is. And it's written in a linear script, which I've already referred to, a linear script of 4 letters, the A, the G, the C, the T. And this linear polymer of DNA is ideal for storing information. And the encoding letter bases are sort of turned inside the helix and sort of protected and inert within that helix, not completely inert, but protected and not chemically active. Now, if it was just stored there and couldn't do anything, it wouldn't be useful. But this information is turned into chemical action by being transcribed and translated into protein. And that's what I think is so extraordinary about this system in life. It's built on carbon-based polymers which have both the ability to encode information in linear structures and the ability to translate that information into chemically active polymers, which are the proteins and enzymes that do the work of cells. And information is central to all of this. The second example I want to talk about with information is regulation. And that is making complex systems working together as a whole with purpose, that is achieving an end for the cell or the organism. You need regulation. Now, I don't know if you're like me, I don't understand any machines basically sort of built after about 1910, okay? But I understand most machines that are built before that. And this is an invention it's called a governor. It was applied by James Watt to regulate steam engines, but it was used prior to that to regulate windmills, I believe. And it's very simple. As that engine goes faster, those balls swing out, and they close the valve and shutting off the steam. And as a consequence of that, the engine slows down. And then, of course, it's switching itself on and off, depending on the speed of the machine. And this leads to homeostasis, which maintains a constant speed of the steam engine. You don't need somebody turning it up and down all the time. It's a governor. It's an example of what we now would call a negative feedback loop. And it's shown here in the context of a metabolic pathway in a cell. And you see at the top there, A is turned into B, is turned into C, and C inhibits A to B. So when you get too much C, it switches off A to B, so the amount of C goes down. Beautifully simple. It's a negative feedback loop. You can have a positive feedback loop, which does completely the opposite, of course, where C speeds up A to B, so once you flip that switch, it keeps going. So then it's a switch rather than homeostasis. Now, this was first demonstrated by some brilliant French geneticists, François Jacob and Jacques Monod, studying gene regulation of sugar metabolism in bacteria. And we now know that metabolism and all functions that go on in living cells are built up of modules of this sort. And these modules can act in different sorts of ways. They can generate switches, timers, toggles, oscillators, translating chemistry now into informational modules. So you see how all of this is linked together. A metaphor for this is this one, as an electronic circuit. I stole this from an EMBO journal paper, and some wit had put on all these oncogenes here as sort of part of a cellular network, which is hardwired, if you could believe this particular metaphor. However, I think a better metaphor is not hardware, but actually wetware, because life's chemistry, which is connected by diffusion of chemicals in liquid, can be rewired. As well as managing the information, you can actually make new pathways, so it's more versatile. And I just don't think we've yet fully understood how that all works. Now, I'm moving to my fifth idea, I'm sure you're pleased to hear. But there is a bit more after that, so if you're getting tired, I recommend deep breaths, okay? This is evolution by natural selection. Biology is a beautiful idea, how living things came about. It's an idea mainly due to Charles Darwin, not entirely. Has two parts. The first part is life evolves over time. That was not Charles Darwin's idea. I'll mention that in a moment. And the second idea is a major mechanism for that evolution is natural selection. And even there, Darwin wasn't completely alone in proposing that idea as well. This is Charles Darwin on the right, and on the left is his grandfather, Erasmus Darwin, a very interesting and eccentric person. And Erasmus Darwin was one of a number of scientists in the 18th century. Look, we're back again in the 18th and 19th century again. You know, what are we doing? I ask you again, what are we doing? So Erasmus Darwin was a colourful character. He was a scientist. He was a doctor. He was a radical politician, a Republican. He wrote all his research, or most of it, in poetry. It's in rhyming verse. I mean, if you read it, I have read some of it. I mean, it's barking mad, but it is sort of interesting. He was a major proponent of women's education and a great conversationalist, despite having a dreadful stammer, so he could hardly speak. But he also argued for the evolution of life, suggesting simple formless mollusks could evolve into complex animals. It was Charles, however, who had a mechanism for how that might come about. And this was the idea of natural selection. And he suggested that the evolution came about through the change of one species into another. And he produced this diagram here, which is a tree of life, the first tree of life, which you can see all at the top is the existing life. Beneath it is all life that's become extinct. And then he has all these letters at the bottom because he was struggling as to whether life came about by more than once, or did it come about in several times? Now, we have no idea how life came about, which is a big mystery to us. But because the chemistry is so similar, that argues that life probably came about once, at least what has survived to today. Now, what was his idea about natural selection? It's a consequence of populations of a species exhibiting variations caused by inheritable genetic changes. So it's linked to the idea of the gene. These can be caused by mistakes when DNA is copied, or if the DNA is damaged by external sources. And if these variants influence the organism so they reproduce more, then that particular genetic variation will gradually spread through the population, because the organism that has it is reproducing more than those that do not. And so over time, such variations which are favourable to the survival, or to be more precise, the reproduction of that life form, will gradually take over the original population and could lead to a new species, better adapted and more successful for survival and reproduction. He came to this point of view in part by study of finches on the Galapagos Islands during the visit of when he was on the Beagle, which went round the world. He spent 5 years on it. And you see how the finches to the left there, some finches have big beaks that could break nuts, others thin beaks that could catch insects under the bark of the trees, better adapted for their environment. And because the Galapagos are volcanic islands, they hadn't been there very long, and he thought maybe a primitive finch got there and then evolved into all these different finches specialized to their different environments. An analogy is what I've got there on the right. I stole it from a book, of course, and these are different pliers which are all similar but designed to do different things. And the reason I put this up is, on the left, they've been formed by natural selection, and on the right, they've been intelligently designed by human beings. So you get a similar outcome, if you like, by utterly different ways. And on the right, we have a designer or creator, and on the left, I argue, we don't need a designer or creator. But that is, of course, still contentious for some even today. Now, as I've already explained, for natural selection to take place, you need several things. First, reproduction. Second, a hereditary system. Thirdly, a hereditary system that exhibits variability upon which natural selection can work. And I want to now link that with two of the ideas I've talked about, which is the cell and the gene. And I want you to imagine a single-celled life form which has a brown cell coat. And the color of that coat is determined by a gene in its chromosomes. So this is very simple, right? I realize I've been talking to you for a long time, so I thought I'd better keep it pretty simple. Imagine, as that cell divides and copies its DNA, it makes a mistake. So sometimes that cell coat gene is altered, and now the cell makes a red coat, as you can see down there on the bottom right. Also imagine that red-coated cells survive better. Perhaps they do not taste good to a predator. And as a consequence, the red cells replace the brown cells in the population, and evolution by natural selection has taken place. And it's built on the concept of the cell and the concept of the gene, and that leads to evolution by natural selection. So these great ideas are all linked. Now, it's a profoundly important idea, natural selection, in biology, and it's going to be central to my definition of life, which I'm about to come to. But before I do that, I want to say goodbye to Darwin, all right? Here he is. This is Charles, now old, and this is the last sentences in his book, actually, The Origin of Species. I read it to you. It's beautiful language. Why can't we write papers like this anymore? You know, you submit papers, you write anything interesting or a bit flowery, scrubbed out by the reviewers and the editors, you know? And, I mean, we're losing the plot, I think. Right. whilst this planet has gone cycling on according to the fixed law of gravity, notice how he refers to the physicists, fixed law of gravity, to Newton, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. It's just great sentences. We just have to start writing like this again. Now I'm going to turn to the title of my talk. What is life? I'm going to pull on the 5 ideas and give my shot at trying to answer that question. I want to define some general principles that will give us deeper insight into what life is. And I'm going to summarize those principles, but I must warn you, it isn't a neat dictionary-like definition. So the first of these is living things are independent, bounded physical entities, and the basic unit of life on Earth is the cell. I emphasize that to deal with the second law of thermodynamics, and I don't want to talk about computer games, okay? Because they're not independent, bounded physical entities. Cells have these characteristics. They're separate from the environment in communication, and they generate order and organization, and multicellular organisms are aggregates of cells. But if we want to understand life, let's understand cells first, okay? That's the first thing we've got to think about. Second, living things, cells, are chemical, physical, and informational machines. All 3 of those things, chemical, physical, informational machines. They construct a metabolism that allows them to maintain themselves, to grow, and to reproduce. All the things that we see life actually does. Third one, and this is the clever idea, first said by Hermann Müller, an American geneticist in the mid-20th century. And he argued that living things can be defined as some entity that possesses the ability to evolve by natural selection, because that way you can generate the diversity of life adapted to its environment. And to be able to evolve, as I've explained to you, you have to be able to grow and reproduce, and you have to have a hereditary system that determines how you operate, which exhibits variability, which comes about in life as we know it on our planet by variability in the DNA sequences. And these, for me, are the basic principles that you have to take account of if you want to understand life. Indeed, I'd argue that these principles define life, and any entity which operates according to them can be deemed to be alive. Now, should we ever encounter life elsewhere in the universe, and I suspect it would be based on similar fundamental principles. The details could be different. It could involve different chemistries, perhaps chemistries not based on carbon, but other polymers, I would suggest. And indeed, for that life to have long-term information storage, I think it's likely the chemistry does need to be based on polymers, because information's written in that sort of way. And that's a digital information storage device. I should also say that living things need energy. I haven't talked much about that, to power the processes of life. That can come ultimately from the nuclear reactions within stars that produce light and heat, or from geothermal energy within planets. And we know that both of those things occur. Let me just talk a little bit more about polymer chemistry. I'm just emphasizing what I've said before. Life on Earth is based on carbon polymer chemistry. The polymers produce nucleic acid information storage devices, and that brings about protein, chemical, and physical machines. Let me repeat. DNA, pretty chemically inert. Protein, wonderfully chemically diverse. Both based on polymers, both related to each other. Life is based on information management, with information stored in that linear form. And living things on Earth are related by common descent, and are closely interconnected and dependent one upon another. Just to recap, the double helix is the way we store information, keeping that storage in the middle. The proteins have a chemistry where you have the amino acids with great varieties on the outside of the protein chain. They're exposed, if you like, to the rest of the world. I frankly can't imagine a more elegant solution. Do you know that? I mean, somebody got it right. Different configurations of linear carbon polymers producing this chemically stable information storage and also highly diverse chemical activities. It's completely extraordinary. And I speculate, that is why I speculate it'll be something similar in organization wherever it may be found in the universe. Maybe the chemistry is going to be different, could be silicon or something, as the writers like. I don't know, but I think the principles there are clear. Now, when thinking about life, it's tempting to try and draw a very sharp defining line between life and non-life. And in fact, there is a sharp dividing line. Cells are clearly alive, and rocks are clearly not alive. But then we have the knotty problem of viruses. Is a virus alive or not? Well, let me just give you my answer to that. You may not find it satisfactory. Viruses are chemical entities. They have DNA or RNA as a genome. They have a protein coat, and they take over the metabolism of cells that they invade. They can evolve by natural selection, as we've seen with COVID, Delta variant that we're all struggling with today. Thus, they pass Muller's test. They pass Muller's test, but they cannot reproduce themselves. The only way they can reproduce is by infecting cells of a living organism and hijacking those infected cells. And so some biologists have concluded that that strict dependence on another living entity means they're not really alive. Now, that's quite a compelling argument, but it's important to remember that almost all other forms of life, including our cells, are also dependent on other living organisms. For example, we can't make certain amino acids which are made by microbes that sit in our gut. We eat the food. Our food is all other living organisms, plants or animals. Some life forms are a bit more independent, like cyanobacteria, for example, but we are all part of a complex, interacting ecosystem. Viruses are at one end of that extreme, occupying other living things, but all of us are, to some extent, as far as I can work out, dependent upon other living things. There's a whole spectrum of it, and we need to be aware of it. So I don't think viruses are quite as different. They are obviously different, but not quite as different as you might initially start with. Another way out of the conundrum, which really is sophistry, is to think about viruses as being alive when they're inside the cell and dead when they're outside the cell. But I do accept that's sophistry. Now, this is quite a broad perspective on life, this interacting system. It gives a richer view of the living world. Life on Earth belongs to a single, vastly interconnected ecosystem incorporating all living organisms. And that connectedness, it's fundamental, not only comes from a deep interdependency, but also from the fact, I'll remind you, that all life is genetically related through its shared evolutionary roots. So we're all connected in time through evolution, today through interaction. And it's this perspective of deep relatedness and interconnectedness that has obviously been championed by ecologists for the last 2 centuries. Humboldt, back to the 19th century, made exactly that argument in Germany, although it was from his visits to South America. So this interconnectivity is core to life. Here we have, oh, that was a virus, I should have shown you that. Back to Darwin's tree of life. Now, the organisms that live today on Earth are part of this shared family tree, and they are truly astonishingly varied. We all agree. But their variety is frankly outshone by their similarities, because we are all very similar. We are built on the same chemistry, and we are built on the same principles that I've just described to you. And that suggests to me that life as it is on Earth today did just arise once. I mean, we can't fully argue that, but that's what I think. It doesn't mean that there weren't several life forms, but the one that we now have survived once. So how did it start? Well, a real problem is a chicken and egg problem. Is it DNA and genes or proteins that came first? And those who think about this, and I've never done any work on this, think actually the answer is that RNA is the solution, because RNA can both encode information and also has enough chemistry to contemplate it could be functioning. And I think I would agree with that. That's the place where I would start. I'm going to come to my last sentences, okay? And I go a bit technicolor now, okay? So do forgive me. Now, the universe is unimaginably vast. So by the laws of probability, it seems very unlikely that across all that time and space that life has only come about once here on Earth. Whether or not we ever meet that other life is a different issue completely. But the vastness of the universe just makes you think there has to be other life forms out there. If we ever do confront it, I'm sure it is likely to be, now that isn't quite right, I think it is likely to be, a self-sustaining chemical and physical machine built around information encoding polymers that have been produced through evolution by natural selection, which is the summary, really, I can have of life. Now, our planet is the only corner of the universe where we know for certain life exists. The life that we are part of on Earth, as I've said, is extraordinary. In spite of its diversity, we're making sense of it. We have a growing understanding of that life, and it can improve the lot of humankind. But it goes further than that, I would say. Biology shows us that all living organisms we know of are both related to each other, including us, and closely interacting, including with us. We are bound by a deep connectedness to all other life, to bacteria, to fermenting yeast, to flitting butterflies, yellow butterflies, and to every other member of the biosphere. We are life's great survivors, the latest descendants of that single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. It really is extraordinary. Now, as far as we know, we humans are the only life forms who can see this deep connectivity and reflect on what it means. And I would argue that that gives us a special responsibility for life on this planet, made up as it is by our relatives. Think about that. It's occupied by our relatives. Some close, many distant. We need to care about it, we need to care for it, and to do that we need to understand it, which is why what I've tried to do in this lecture, and if you're interested in it, I've also written a book which is here, that you can find in French and German, as I know this city is divided into both those forms. Thank you very much for listening to me.